Hello everyone, welcome back to our channel Fox Science. Today we will discuss about the translation elongation and translation termination that would be the ultimate part of the translation process. So let's get started. Elongation process is the three-step microcycle. First step is the decoding. Decoding means the ribosome select and bind the amino acyl tRNA whose anticodon is complementary to the mRNA codon present in the A site. Why A site? Because P site will be already occupied with the initiator tRNA. Initiator tRNA, as I mentioned in the last video, that it will come and always enter into the P site first. Then the second amino acyl tRNA will always enter in the acceptor region or A region of the ribosome. So the second step will be the transpeptidation. This step is conducted by peptidyl transferase enzyme. This is a very important enzyme in translation process, and in bacteria it is encoded by 23S rRNA of 50S ribosome, and in eukaryote it is encoded by 28S rRNA of 60S ribosome. So in transpeptidation process, what happened? The peptide bond between the amino acid present in the P site and amino acid present in the A site is formed by the peptidyl transferase enzyme, and hence the peptide chain, polypeptide chain will start to continue and start to form. And the third step will be the translocation process. In the translocation process, three factors are involved, like EFTU, EFTS and EFG. What does EFTU do? Actually, the EFTU gets activated by binding with GTP. EFTU and GTP form binary complex, then it attached with the amino acyl tRNA, and then this group gets attached with the acceptor region or A region of the ribosome. And EFTS is the GEF, means guanine nucleotide exchange factor. When EFTU need to become activated, it dissociate the GDP and GTP gets bind, like vice versa. And EFG causes the ribosomal translocation in 5' to 3' direction. So if we discuss in a single slide the entire elongation process, here you can see the amino acyl tRNA along with the EFTU GTP gets attached with the activated site, and in the P site already the initiator tRNA with the methionine is present. Then the peptide bond is formed by the peptidyl transferase enzyme and the initiator tRNA gets vacant. Then ribosome will translocate and the charged amino acyl charged tRNA will enter into the P site, and the vacant tRNA will get outside with the exit or E region, which is present in the larger ribosomal subunit, and hence the A site again will become vacant to accept newly charged amino acyl tRNA. So hence the entire elongation process continues, and until the stop codon reach. We have three stop codon, UAG, UGA and UA, like UGA, UAA and UAG. These three stop codon, until the ribosome reach to this stop codon, the elongation process continues. When it reaches the stop codon, in case of prokaryote, three factors are involved: RF1, RF2, RF3, means release factor 1, release factor 2 and release factor 3. So release factor 1 recognize UAG and UAA stop codon. Release factor 2 can recognize UAA and UGA, and release factor 3 helps in the dissociation of RF1 and RF2 after the termination process. So first RF1, RF2 actually contain one termination sequence that is called GGQ sequence. So they bind with the A site, and then it terminate the entire translation process, and RF3 along with GTP, it helps in the dissociation of the RF1 and RF2, hence the translation process is terminated in the prokaryote. In case of eukaryote, the factor which helps in the release termination of the translation, that is the ERF1. It can recognize all the 3 stop codons, but it can terminate the translation. It cannot helps in the dissociation of ribosome. For the dissociation of ribosome, RRF or ribosome release factor is needed. It dissociate the ribosome in a GTP dependent manner. So this is the elongation and termination process of translation in eukaryote and prokaryote. If you have any further question related to this video, please do comment in our comment section. If you like this video, please do like, subscribe and share. Thank you so much.